Good morning all. Today we are going to see chapter 6 part 4. So up to last class we discussed about the transcription process in our central dogma. So in this class we are going to see translation process. Before getting into the translation process, I am going to give you the overview of what is genetic code and what about mutation. Okay. So genetic code, we know that central dogma, DNA to RNA, RNA to protein. Okay, so that was discovered by Watson and Crick in the year 1952. So at that time itself, this genetic code was determined by the scientist who is said to be George Camo. Okay, so he is the one, he concluded how to write the genetic code. Okay, genetic code means the information which will code for a particular protein is said to be the genetic code. Okay, so it is made up of nucleotides. So what he told means, we know that there are 4 bases that was discovered by Watson and Crick. Okay, so this George Camo, he said that 4 nucleotides are there, that is adenine, guanine, cytosine and thymine. So these 4 nucleotides only has to go code for our 20 amino acids. Okay, so how that 20 amino acids can be coded? Okay, so he is basically a physician. Okay, so he did that so many experiments, so and he concluded that by means of permutation and combination we can make that 4 nucleotides can be made to 64 amino acid sequences. Okay, so he said that the 4 cube which gives us 64 codons, these 64 codons will code for our 20 amino acids. This was concluded by George Camo, right? So this experimental analysis was carried out by 2 other scientists also. One is Hargobind Khorana and another one is Marshall Nierenbergs. So what these 2 scientists told means, they did so many experiments and they also concluded that this is the best combination. Okay, and they gave us the checkerboard for the genetic code. Okay, how to write the genetic code in an universal manner. Okay, so how can we write this genetic code means, we know that it is made up of 4 nucleotides. So 4 cube we are getting a 64 codons, right? So we have to write like this. So in this left side we have to write individual nucleotides, and on the top of this you have to write the nucleotides individually. So coming to the right side, you have to give the 4 nucleotides based upon each columns. Okay, so based upon this we can write the nucleotides as follows: U from left, U from top, and we can write this U from here. Okay, so I will write you the sample, then you can understand it clearly. So see here in this second column, so I am writing U, the next C has to come, then first U has to come. So like this you have to write U, C, then second C has to come, U, C, A has to come. Okay, U, C, G has to come. So like this we can fill up this whole tablet column. So you will get a 64 codons. Okay, so this is the genetic code checkerboard. Okay, this is an universal method. So all over the world they are following the same principle. So there is no confusion. So coming to the salient features of our genetic code. So they said, we know that there are 64 codons, right? So among these 64 codons, only 61 codons will code, right? So based upon that we are dividing it into, one is said to be the start codon and one is said to be the stop codon. This stop codon also we can say it as nonsense codon. Okay, which will not code for anything, which will not code for a protein molecules. Only the 61 codons will code for the protein molecules. Among that 61, we are saying one is said to be the start codon. The start codon is AUG. You have to remember the 4 codons alone: UAA, UAG and UGA. In simple we will say it as amber, ochre and apple. Okay, so these 3 codons are said to be the stop codons and AUG is said to be the start codon. Okay, so I told that these 3 won't code for any amino acids. Okay, so this start codon AUG, which will code for an amino acid methionine. So this is the first codon which will be present in our translation process. Okay, so that is why we are saying this as a start codon. Why we are saying these 3, amber, ochre and apple, as a stop codon means these 3 are said to be the terminators. Okay, terminators means last transcription also we have studied, it will end the process of translation. Okay, for translation these 3 codons, if it is arising in the mRNA, means the translation process will get over. Okay, so among these 64 codons, 1 is said to be the start and 3 is said to be the stop codon. Coming to that code, these 64 codons are degenerate. Degenerate means nothing but one codon can code for different amino acids. Okay, so see here example, this UUA. This UUA is responsible for coding an amino acid phenylalanine. Okay, the same UUU can code for some other amino acids also. See here, this UUC is coding for an amino acid, same phenylalanine. UUA and UUG, both is coding for an amino acid lysine. Okay, so you can see, so these 64 codons can code for a multiple amino acids. That is said to be the codes are degenerate. The next one is, we should not read the code with the help of any punctuation marks. For example, if you are having AUG, CG, TTT, means you should not put any comma or any full stop or any dash in between this. Okay, this is completely wrong. We have to read it as such, AUG, CGG, TTT. Okay, we should not give any punctuations in between them, and we should not leave any gap between them also. Okay, this code is said to be the universal. These are the properties of our genetic code. Okay, so coming to the mutations, we have read in the last chapter itself point and deletion. Okay, so here this insertion as well as the deletion will change the frame itself. I will give you an example. See here, Ram has a red cap. Okay, so this is the normal sequence. So consider this as the DNA sequence. Okay, this is the normal one. So here, instead of this R, if B is inserting, okay, we know that insertion means addition of nucleotides to the DNA molecule. That is said to be the insertion. Deletion means deletion of a single nucleotide from the DNA molecule is said to be deletion. So I have given an example. One is insertion and another one is deletion. Okay, so in this frame, so in this sentence, one P is inserting. So we know that codons will always be in 3 nucleotides. We have to read it like this only. AUG is coding for one amino acid, CG is coding for another one amino acid, TTT for another one amino acid, right? So only it has to contain the 3 nucleotides. So if B is coming and inserting means, if you read the code, if the DNA polymerase is reading it, means it will read it wrong, right? Because the insertion of B is there. Ram has red, it has moved outside because one more B is inserted. So the complete frame itself, the sentence itself is changing here, right? The same here also. Here we are deleting this R. Okay, so what will happen? So Ram has, instead of R, this R was deleted. So obviously our RNA polymerase will 3 letter only. So it will take the next letter C and it will read as EDC. So obviously what will happen here also, the protein synthesis will be completely wrong. Okay, so if there is any mutation caused by the mutagen, means the whole process of protein synthesis will be deleted. So this is the basic information we have to know before getting into the translation process. The genetic code, which is made up of 3 codons, that you have to know. And another one important thing is start and the stop codon. Okay, so now getting into the process of translation. So this translation, so before we have studied replication and transcription, right? So replication as well as transcription, both are very easy process because the same nucleic acids only will be coding. But coming to the case of translation, here it is a little tedious process. Why? Because here amino acids are involving, tRNA are involving, as well as ribosomes plays an important role. Okay, so here the complete set of nucleotides is going to converted into a protein molecule, a whole set of protein molecules. So this process will take place by means of various steps and involving the various cell organelles also. We know that up to that transcription process, it will happen inside of the nucleus. Okay, so after this transcription process over, your mRNA will leave the nucleus via nuclear pole and it will enter into the cytosolic region. So we know that this is the cytoplasm, it will enter into the cytosolic region, right? So here in this cytosolic region, wherein your tRNA will be present as well as our ribosomes will be found, okay, and amino acids will also scattered only in the cytoplasmic region, right? So these are all the requirements for our translation process. So the first process in translation means what? The conversion of mRNA molecules into a complete protein molecules by forming the peptide bonds. That is said to be the translation, okay. So here in this first process, the amino acid which was scattered in the cytoplasm has to be activated first, has to be charged first, okay. That is the first step, activation of amino acids. So for the activation of amino acids, we are in need of ATP molecules, okay. The energy is needed for the activation of amino acids, and particular enzyme is needed. That enzyme is said to be the amino acyl synthetase, okay. So we can simply say it as E, okay. This E represents amino acyl synthetase. So after this amino acid present in the cytoplasm along with our ATP plus our enzyme, you will be getting a complex AAAMP-E complex. I will explain what it is. So this activated amino acid, now the amino acid is getting charged. Why? Because here you are having this triphosphate. So this two phosphates will be released outside, and you will be getting monophosphate, amino acid monophosphate, right? So ATP here converted into AMP. Triphosphate has converted into an monophosphate along with the enzyme complex. So these complete set, we are saying it is amino acyl. Amino acyl is nothing but an activated amino acid, and the two phosphorus got eliminated. Clear? So this is the first step. Only one exception is there. In case of our start codon, we know that start codon is nothing but our AUG, which codes for an amino acid methionine, right? So only for this methionine, this amino acid charging is little different. Why? Because instead of this amino acid synthetase enzyme, here we are saying the name as F-met, that is formulated methionine. Why? Because in order to avoid the mutation process, this methionine was charged by different means, that is formulated methionine. We are saying only for this amino acid F-met. Remaining all the amino acids, it will follow only this process. Clear? So after this activation of amino acid, so what will happen? Our tRNA molecule will go and carry the amino acids. So before getting into the transfer of amino acid to the tRNA, I want to explain you the structure of our tRNA molecule. So from our lower class, we have studied this clover leaf model, right? So this is the structure of our tRNA molecules. We know that a tRNA is a single strand, okay. This is the single strand. We will be having 5'N and 3'N. So this is the normal structure. This converted into an clover leaf structure, like this you imagine, okay. So 5' and 3'. So if you are seeing the structure under the microscope, it will be visible like this only. So we are having 4 arms, okay. So this is said to be the tiny arm, just leave it. We are having the 4 important arms. Among these 4 arms, only 2 arms is getting involved in our protein synthesis. That is, first is said to be the amino acid acceptor arm. So in this, we are having 5' and 3' region. We know that 3' is having a free hydroxyl group, right? So in this hydroxyl group only, our amino acid present in the cytosolic region, that is our charged amino acids, will come and attach in the OH region of RNA molecules, that is tRNA molecules, and this region, that is down region, is said to be the anticodon arm. Here only our anticodons will be present. So this anticodons will code for the codons in the mRNA. Can you able to follow? So this 3' end is responsible for the amino acid acceptor arm, and this end, that is the down end, which is said to be the anticodon arm. So anticodon, we know that it is anti means it is opposite to codon, right? So it will code the complementary sequence of codon present in our mRNA molecules. Clear? So this is the basic structure of our tRNA. So here what will happen means, after this complex, we are saying this as the charged amino acid. So this charged amino acid is coming and attaching our 3' end. So only our amino acyl will come and attach, and the remaining AMP plus our enzyme will be released outside. Amino acyl synthetase and monophosphate will get released into the cytosolic region. So our tRNA now it is carrying the amino acid with us. So next what will happen? Amino acid was charged and it was carried by our tRNA molecule, right? So the next thing is this tRNA has to go and bind with our mRNA molecule with the help of ribosomes. So now the role of tRNA is finished. Okay, so it has added the amino acid. Next our hero is coming, that is our ribosome. Okay, so we know that the ribosomes are made up of two subunits. One is said to be smaller and another one is said to be the larger subunit. Okay, so the two subunits will not present together. Okay, so unless or otherwise there is a protein synthesis. If protein synthesis is going to happen means then only the larger subunit and the smaller subunit will unite together. Okay, so here in this ribosomes we know this is the smaller subunit and this is the larger subunit. Okay, so what is the role of these two subunits means? In this smaller subunit only our mRNA molecule, which was produced by the process transcription, okay, will come and attach only in the smaller subunit. So this larger subunit you are having a three regions EPA. In short you can say it as EPA. So here we will see one by one. This A region is said to be amino acid site. Okay, so here only our amino acid will come and attach. So this is the larger subunit of ribosome. Three compartments are there, E, P and A. In this A region only our amino acid along with our DNA will come and attach. Okay, so next is P region, which is said to be the peptide site. Okay, see here the peptide bond formation will be takes place. So what is the role of peptide bonds? It will unite one amino acid with other amino acid. Consider this is the two amino acids. So this amino acid has to be attached with this amino acid by means of what? Peptide linkage. Okay, so in this compartment only that process will be takes place, peptidization. Okay, so next is said to be exit. So after the peptide bond formation our tRNA will be coming to the exit region and it will exit outside. Okay, this is the basic principle. So see here only during the protein synthesis our ribosomes will get unite together. Here smaller subunit and larger subunits are united together. Okay, so now see this is the mRNA. Consider this is the mRNA molecule means, so our ribosomes will come and bind like this. Okay, so our mRNA will be as such like this. Only the ribosome molecules will move like this. It will move from A site to P, then it will move to A, again E, again A, P, E, like that the movement of ribosomes will be takes place. Our mRNA will be stand as such. Okay, so only the ribosome molecule will move and it will code for our protein molecules, that is the peptide bonds will be produced and our protein molecules will be synthesized. Coming to this protein synthesis process, if you see the diagram you can able to view it clearly. So this is the smaller subunit. Okay, so the role of smaller subunit is the attachment of mRNA molecule. So here only our mRNA molecule will get attached. So no other role of ribosomes here, no other role for our smaller subunits. So coming to this larger subunits, as I said earlier, here we are having three compartments E, P and A. So first the role is A site. Okay, in this A site our amino acid along with our DNA will come and attach into the, it will code for the anticodons. Okay, so the next the ribosomes will move and it will move to the P site. Once the amino acid is getting attached means the role of P site is to produce the peptide bond. So see here in this diagram, tyrosine, serine, glycine, serine, alanine, everything the amino acid is moving outside. Okay, so by means of this peptide linkage. Okay, so how this peptide linkage happening means by the peptidylation process your tRNA molecules will code for the anticodons and our peptide links will be formed. Okay, so the complete set of amino acids will be formed. So after this amino acid in this E site, that is exit site, here our stop codons will be present. So once the stop codon is there, it will stop and it will move to the next region. Okay, so this is the continuous process. So once all the amino acids are formed by the peptide linkages, so one complete protein is synthesized means our translation process will get stopped and these two subunits, larger as well as smaller subunits, will get moved together. In this also we are having some non-coding regions. So if there is a non-coding region means, I have told already, that is said to be the regulator genes. Okay, so some non-coding regions will also present in behind the start codon as well as the stop codon. Okay, so that will regulate the process of translation. So this is the simple method of translation. So in this translation process, we are having 3-step activation of amino acid. After this amino acid activation, it will transfer to the tRNA. So again the tRNA will carry the transferred, that is charged amino acid. So here the charged amino acids will come, and in the peptide region you will be having the formation of peptide bonds. So after the peptide bonds are formed, it will exit in the exit region. So our tRNA will get separated, amino acids will get separated, ribosomes will get separated, and mRNA will get separated, and they will move to their places. And again the process is a continuous one. So till our death, this process will goes on. Okay, so the cell division will carry out and the protein synthesis will carry out till the end of the life. Okay, so this is the simplest process of translation. So next class we will see how this transcription and translation was regulated, controlled. Okay, so this is the important chapter, that is the regulation of gene expression. We know that gene expression means, so from the DNA our genes will be expressed into the protein, and it will give you the physical appearance to any human beings as well as the microorganisms. Physiological as well as the anatomical functions will be carried out only by means of this gene expression. Okay, so how our body is regulating the genes? It is just a simple method, just like switching on and off of our fan. Okay, so for example, if I am taking the starch material or if I am taking the protein material, means my body has to synthesize an amylase to convert the starch into the glucose. Same protease enzyme has to convert the protein into simple product for the digestion process, right? So for that expression of the protein, that is the amylase as well as the protease, our genes have to be regulated. So if I didn't took starch molecule, means my amylase will not produce, right? That is said to be the regulation. So if I didn't took starch molecules, if my amylase is producing, means so some wrong was happened in my gene. It is not regulating properly. Okay, so even for us, that is for eukaryotes as well as for the prokaryotes, the regulation is very much needed. Okay, so if the regulation is not there, means then mutation will happen, then our normal genes will convert into an oncogenes. It will lead to the cancer, right? So here we are going to see the gene regulation in eukaryotes as well as in prokaryotes, but in your textbook they have given in detail about the prokaryotes, right? First we will see what is the gene regulation, how it is happening in the eukaryotes. We know that eukaryotes contain a monocistronic genes. Okay, so here the single polypeptide chains will be produced, so there won't be any regulations that much when compared to the prokaryotes. So in this case of eukaryotes, so transcription level the gene is regulating. How in transcription level means, we know that from the DNA we are having the hnRNA, which is said to be the pre-mRNA, then only our mRNA is producing, right? So this heterogeneous nuclear RNA which contains some unwanted genes, okay, so that will be deleted while during the transcriptional process. So this is one regulation level. Okay, so another one is processing level. Processing level, we know that in our hnRNA we are having that two sites. One is said to be the introns and another one is said to be the exons. We are having coding as well as the non-coding regions. Okay, so during the splicing process our introns will be removed, we are having only the exon. This is also one of the regulation because our unwanted genes are regulating. And during the transport of this is also one process, that is the transport of mRNA from nucleus to the cytoplasm for the translation process. This was controlled by some of the genes. Okay, if there is a need for translation process, then only our mRNA molecule will enter the cytosolic region. Okay, so at the level of translation, so during the translation level we know that the tRNA, tRNA, ribosomes plays an important role. Okay, so these tRNA will code only for the particular codon, because in this tRNA we are having the anticodon region. Okay, so this anticodon region is also responsible for regulating the genes. That's all about your eukaryotes. So in this eukaryotes the process is very simple, because during the transcription as well as the translation process itself the regulation is happening. Okay, and only the necessary genes are producing. But in case of prokaryotes, so for prokaryotes you can take as an example Escherichia coli. Okay, take a bacteria E. coli as an example for prokaryotes. So here in this transcription process I have told you already, we are having a promoter region, structural region and a terminator region. Okay, so in this promoter region itself our regulation will get started. Okay, in case of prokaryotes. So here we are having regulatory genes. I have told while taking the transcription process itself there is a regulatory genes which is present beyond the promoter region. Okay, so this regulatory genes we can say it as a regulatory protein which will act as a on and off mechanism, that we are saying it as positive regulator as well as the negative regulator. The regulator genes present beyond the promoter region is acting as an on and off mechanism. Okay, so here if you see it in detail, we have given an example, lac operon. Operons means nothing but a set of genes which regulates this transcription as well as the translation process. That is said to be the operon. A set of genes regulates the protein synthesis. Okay, so here this lac operon was first discovered by Jacob and Monod. Okay, so here lac means which is nothing but our lactose. So for this Escherichia coli we have to know one concept. Any bacteria, you can take E. coli or lactobacillus, whatever the bacteria may be, the bacteria has some source for the survival, right? We need starch. So like that the bacteria will uptake glucose molecules from the medium. Okay, if the bacteria we are growing it in an artificial medium means we will supply the nutrient medium. Okay, so in that nutrient medium we will be having glucose molecules. If glucose is present, it is well and good. The bacteria will absorb the glucose moiety as such. For example, if we didn't provide glucose molecules, for example we are providing a lactose. Okay, so compared to glucose, the lactose is a little complicated structure. Okay, the bacteria cannot easily destroy it. If there is an absence of glucose only, the bacteria will go for the lactose in order to survive. Okay, this is nothing but a survival of fittest condition only. So in the absence of glucose only, our lac operon will get activated only in the presence of lactose. Here same like lac operon, we are also having some other operons, that is trip operon, histidine operon, valine operon. These are all for each individual amino acids. Each and every operon is having a set of genes for its own control. Okay, that is why we are saying it is an operon. Okay, so here if you see in this lac operon we are having two genes. One is said to be regulatory gene and another one is said to be the structural genes. Okay, so this regulatory genes we are giving the name I. Don't confuse it with inducer. Okay, so in this lac operon our lactose is acting as an inducer. Our I, that is the gene, regulatory gene, is acting as an inhibitor. You have to know the difference between inhibitor and inducer. Okay, first we will see what is inducer. Inducer means a compound which is responsible for starting any process. Okay, starting means for inducing. If the compound is present means then the process will start, right? So if it is an inhibitor means it will stop the particular reaction. If I is coding for a protein, that is inhibitor protein, means if inhibitor protein is present there, then there will be the termination of that process. Clear? So regulatory gene is said to be I will code for an inhibitor molecule and our lactose is acting as an inducer molecules. So coming to the structural genes, we know that regulatory genes are there, promoter gene is there, and one more thing I left it, we are having one operator genes. We know this operator is also a protein which will regulate the promoter region. Okay, this operator regulate our promoter site. Okay, so three genes, one is regulatory and promoter region and operator region. The function of the operator region is it will control the promoter site. So this regulatory gene will code for an inhibitor our lactose is acting as an inducer. This structural genes, okay, so we are having in lac operon, we are having 3 structural genes. One is said to be Z and another one is Y, third one is A. Here Z is coding for an enzyme beta galactosidase and Y is said to be an enzyme permease and A is said to be trans-acetylase. Now we will see the role of these 3 enzymes. So in the structural genes we are having the first one is Z, which is coding for beta galactosidase. The role of beta galactosidase is it will convert the complex lactose into a simple sugar, that is galactose as well as glucose. Why it is converting means the bacteria cannot able to uptake it as such. Okay, so if it is converted into a glucose means it can easily undergo the process, it will it will digest it easily. Okay, next one is said to be permease. What is the role of permease means it will permeability of the cell, so it will allow the lactose molecules to enter into the cell. Okay, so this permease only giving a pore in the cell wall and our lactose molecules will get allowed. Okay, so for the entry of lactose this permease is much useful. The next one is A gene means it is coding for an enzyme trans-acetylase. What is the role of trans-acetylase means? Before getting into this, I want to tell you one thing. So this lactose was not taken by the organism as such. Okay, we are having one name that is said to be allolactose. So the lactose has to be converted into an allolactose with the help of this enzyme trans-acetylase. Okay, so it will add the acetyl group to the lactose and it was converted into an allolactose. This is the function of trans-acetylase enzyme. So Z gene is for converting the lactose into glucose and galactose and Y gene is coding for permease enzyme which will permit the entry of our lactose into the cell, and trans-acetylase is an enzyme which will add the acetyl group to the lactose and it will convert into an allolactose. Okay, so these are all the enzymes which are present previously in the cell itself. If lactose is coming means these 3 enzymes will be very active and it will recognize the lactose and it will convert into allolactose and bring to our repressor side. Okay, so this is the basic genes present in our lac operon, regulator as well as the structural genes. Now we are going to see both the cases. What happens if there is an absence of lactose and what happens if there is a presence of lactose. Okay, so first we will see in the absence of lactose. If there is no lactose in the medium means there is no need for these enzymes, right? Beta galactosidase, permease and trans-acetylase. Okay, so if there is an absence of lactose means we know that our repressor molecule, that is I, is representing the repressor gene we have studied now. So this I will undergo the transcription process and produces the repressor mRNA, and this mRNA will undergo translation and produces the repressor protein. So what will happen means if there is no lactose means there is no need for the activation of these structural genes, right? So our operator gene will be very ready for the transcription process. But though there is an absence of lactose, what will happen means our repressor molecules, we are having some particular binding sites. Okay, so this repressor molecule will go and bind with the operator, okay, and prevents the transcription process. So there won't be any synthesis of Z, Y and A. Here the mechanism is off. Okay, the lac operon is off. If there is an absence of lactose, our repressor molecules will go and bind with the operator gene. Just like a lock and key model, this enzyme will go and bind to the operator. So if our RNA polymerase is coming means, so this repressor molecules will be binded here, right? So this will, our RNA polymerase won't be allowed after this operator gene, because our this repressor protein will go and bind with the operator and it will block the region. So there won't be any process of transcription as well as the translation. So this is one kind of regulation in the absence of lactose. Now we are getting into the presence of lactose. If the lactose is present in the medium means what will happen? The lactose will enter into the cell through the enzyme permease, and our trans-acetylase enzyme will convert the lactose into the allolactose. Here we know that the lactose is acting as an inducer. So what will happen means the same process here, the I is coding for a repressor repressor gene, it will code for the repressor mRNA by the process transcription, then it will move to the translation process and produces an enzyme repressor. Okay, so here note this thing, this is the main important thing. So here in this repressor gene we are having a particular region, right, we have studied here. So this region will go and bind to the operator if there is an absence of lactose, but here what is happening means our inducer will go and bind with the repressor molecule, okay, and it will block the repressor molecules to bind from operator. Now our operator is very free, okay. So if our RNA polymerase is entering means it will freely move, okay, and freely move, and it will convert the Z to beta galactosidase, Y to permease, A to acetylase, okay. So our, if the lactose is present, the process will be induced, transcription and translation will be happening, and these enzymes are present and it will convert the lactose into glucose moiety. So what will happen? Obviously if the glucose is present means then the bacteria can able to eat it and it will survive. So this is the important part of regulation. So here in the absence of glucose or absence of sugar, what is happening? The repressor moiety is blocking the operator, but it is vice versa here. If the lactose is present means the lactose will go and bind to the repressor and it will block the binding with the operator region, okay. So obviously our operator is set free and our RNA polymerase is also set free, so the complete protein synthesis will be takes place and these enzymes are produced and our E. coli can able to survive even in the presence of lactose also. Clear? So the next part I will put in PowerPoint presentation, then it will be easy for you to understand human genome project as well as the DNA fingerprinting. Thank you.